Welcome back, everyone. Next up, we have Regen BioPharma Inc. It trades on the OTC under the symbol RGBP, and it's focused currently on mRNA and small molecule therapies for treating cancer and autoimmune disorders. Please welcome its president and CEO, David Koos, and senior scientific consultant, Harry Lander. Welcome back, gentlemen. How you doing?
Oh, really good. Glad to see you.
Thanks for having us.
All right, Take it away.
Okay, so today's presentation really a follow-up to our first presentation. We have received quite a few questions that have popped up since the first presentation, so much so that we have an abundance of items. And the goal here today is to really update you on DuraCAR, which is one of our 3 preclinical therapies that we're in the midst of. So the actual questions, a lot of the questions we received, I expect to address in a letter to shareholders, which we'll make public on both our Twitter and also up on a news release. So I would encourage you all to send in all the questions you would like to see addressed, and we'll try to get to most of them in that shareholder update. With that being said, I'm going to turn it over to Harry Lander, who's going to discuss DuraCAR.
Thanks, David. And I'm very happy to be back and talk a little science. If you recall, this was our existing pipeline, and today's I wanted to focus a little bit on DuraCAR, which is one of the newer CAR T therapies that we're working on. And as David mentioned, we have multiple cell therapies ongoing in the preclinical level. And we're very excited about DuraCAR. I would like to describe it a touch. So as I mentioned in the earlier presentation, so DuraCAR takes advantage of our patented technology to inhibit NR2F6, one of the checkpoints that are in T cells, using shRNA technology. And we believe that by inhibiting NR2F6 via shRNA in these CAR T cells will have long-term durable effectiveness. They will not be inhibited, and I'll get into it in a little bit. So we want to make these CAR T cells much more effective at killing solid tumors. Killing solid tumors has been a problem for the CAR T space, whereas its effectiveness in liquid tumors has been superb for leukemias and B-cell lymphomas, for example. But the CAR T space has not really been able to attack solid tumors for several reasons. But I just thought I'd briefly show you, many of you probably know, the treatment process for CAR T. So a cancer patient who requires CAR T therapy, their T cells are removed and brought into a laboratory where they are actually reprogrammed. So these CAR molecules, chimeric antigen receptor molecules, are put into their T cells. The T cells are then now programmed to attack their cancer cells. And before the CAR T cells are put back into the patient, they have to be expanded. So millions and millions of these CAR T cells are expanded in the laboratory, and then they're put back into the cancer patient. At a simplistic level, that's what goes on. If we dig down a little bit deeper, and this is a little bit of a complicated slide, but sort of gets into the science of what we're doing with DuraCAR, you'll see the T cell is at the bottom, the tumor cell is at the top, and these chimeric antigen receptors, CARs, are what we are putting into our DuraCAR. And it has an outside part that sticks outside the T cell, and that allows it to bind to the target tumor. And it has an inside portion, and the inside portion consists of these signaling molecules, so that when the CAR T cell recognizes that it bound to the tumor, it then signals inside the cell and says, okay, let's kill this tumor, let's replicate, let's get very active. And there was a first generation, second generation, third generation of these CARs adding signaling molecules inside the cell to make them more and more active. And we are actually, in our case, we're using a 4th generation where we not only have these signaling molecules, but we also have a transgene that, as I mentioned before, it's the shRNA that inhibits NR2F6. NR2F6 has been a target of Regen for a long time. It's a very important checkpoint, immune checkpoint. Checkpoints are molecules in or outside of immune cells that stop the T cells from becoming active. And by inhibiting this inhibitor, you then release the T cells to become very active and destroy tumors. And so we are now using this technology inhibiting NR2F6. And the scientific rationale and targeting solid tumors in particular is that, as I mentioned, solid tumors don't have a very good track record of being attacked by existing CAR T therapies. There's 4 main reasons for this. 2 of the biggest ones are what's called exhaustion and the fact that immunosuppressive factors are secreted. So these solid tumors exist in a microenvironment, and immunosuppressive factors, both by the tumor and other cells in this tumor microenvironment, shut down the CAR T cells or regular T cells. And in addition, there's a related phenomenon called exhaustion, where these CAR T cells, even when they do get in, by signaling continuously, they shut down. So the solid tumors, as I said, are very difficult to attack with existing CAR T. We believe that by inhibiting NR2F6 in our CAR T cells, they will not be prone to exhaustion because this checkpoint has been released, and they won't be suppressed by these immunosuppressive factors, 2 very big and important factors in the CAR T space. So we're beginning a series of scientific experiments to address that, and I just wanted to show some data we recently generated in partnership with our contract research organization, where we, on the left side, you'll see these 2 panels. The left-hand side are cells that have not been transfected with our DuraCAR, and on the middle panel has been. And you see that rectangle in the middle, and you see the rectangle now has a lot of those blue and green dots in it. And that's showing that CD19 is being expressed on the outside of these cells. So that was a very exciting finding. And we expected it, but it's, you have to do the experiments. And then on the right-hand panel, equally exciting is that it shows that we are simultaneously expressing the shRNA for NR2F6. So the NU, those are the null cells. They are not expressing our DuraCAR vector. And on the right-hand side, that big black bar, you can see that that's the NR2F6 shRNA being expressed. So it's working in our, as we expected, which was, again, very, very positive for us. So that's where we are with DuraCAR. And again, you can see it here at the bottom of our pipeline, and it's capitalizing on our existing intellectual property. So I just wanted to, in the limited time we have, to show you that, and hopefully maybe we can get some questions from the audience. I'm going to hand it over to David. Thank you.
All right. So our first question, I believe, was looking at what the next steps are with DuraCAR. I think, Harry, do you want to address that?
Sure. So as I was getting at, we did these phase 1 in vitro studies just to show that the chimeric antigen receptor was expressed, and that the shRNA for NR2F6 was also expressed in the same cell, and it was. So that was terrific. The next step, we need to then put this vector into T cells and create these CAR T cells. And again, make sure that these T cells are expressing the CD19 and expressing this NR2F6 RNA. And then make sure that the NR2F6 gene is knocked out. So that would be the immediate next step for DuraCAR. I think it'll take about I don't know, somewhere three months or so to get those data. And then the step after that, the third step would be to then take these CAR T cells that's truly the DuraCAR now, and test them initially in vitro to see if they are more effective at killing tumors, and to test it, to make sure that they see that they're more persistent, that they don't undergo exhaustion, and then of course we would move to the in vivo models as well.
Excellent, excellent. Now, the follow-up question is, somebody wrote, I'm trying to understand DuraCAR better. It seems like a completely new way to treat cancers. Is that true?
I wouldn't say it's a completely new way to treat cancers, because CAR Ts now have been around since 2017 was the first approval. So they've been around for 5 years, but it is definitely a completely new type of CAR T cell where we're inhibiting something inside the CAR T to allow it to be even more effective.
Okay. And the final question we're going to take today is, can you speak a bit more on how you generated your data on DuraCAR?
Yeah, sure. So we use a contract research organization. So these are large organizations, and I mean like very large organizations with tens of thousands of employees in some cases, which do contract research for all the big pharma, for big biotech, and they have a vast amount of experience. And so we design the experiments in-house. We have our own intellectual property, but then we contract it out to these CROs. And to me, there's a huge advantage to using CROs because they have a lot of experience in running all kinds of these types of experiments, and they can give feedback. So when you have 5,000 scientists working at your organization, you're going to know what's commonly done, what the problems are. So we get benefit from their knowledge, and they, we don't have, or David doesn't have the overhead. Maybe you want to speak to that, David, but we don't have the overhead of having all those laboratories and the scientists at Regen.
Correct. Yeah, that's a big feature for younger stage companies, not to get burdened with a huge overhead with all kinds of scientists on staff, building, and all the expenses with running a lab. Okay, that was our last question for now. I know that we have a lot of questions that we have received, and I would like to kind of bundle those up in my year-end CEO's letter to the shareholders. If you have any other questions you would like to see addressed, get them into me as quickly as possible. You can send them to me via the website email, and we will try to get all these tied into one good response in the shareholder letter. I appreciate your paying attention to our seminar. Anna, I think was really good, and I appreciate the opportunity to speak to you guys.
All right. Yes. Thank you, gentlemen. We do have lots of questions from our audience, but we'll send them directly to you if you prefer answering in that way.
Perfect. Hey, appreciate it.
Okay. Wonderful. Well, thank you for this update, and we look forward to seeing you on again in the near future.
All right. Thanks.
Thank you.
All right. Thank you, guys. Okay. Stay with us, everyone. We'll be back in just a few minutes.